Hi everyone, today's screencast is on photosynthesis. So this is B1 revision and the screencast will cover the topic of photosynthesis, what the products are used for. We'll also be talking about limiting factors and doing some calculations as well. So the learning objective today is to know what photosynthesis is and what the products are used for. To start us off, I'd like you just to complete the retrieval practice. So pause the screencast, have a go, and then unpause when you have finished. Okay, so number one, name a non-communicable disease. You could have said red-green colour blindness. You could have said that diabetes is a non-communicable disease. You could have said cancer. And there are other examples. So something that is not passed by pathogens, something that is not spread from organism to organism. Okay, question number two, which pathogen causes the flu? That is a virus. Number three, what causes red-green colour blindness? It is caused by your genes. So you would inherit genes from your biological parents that would cause you to have red-green colour blindness. Number four, what happens if a blood clot happens in a vessel leading to the brain? It would be a stroke. And number five, which type of blood vessel has thick muscular walls and carries oxygenated blood? They are the arteries. Okay, make sure you have assessed those, make sure you've marked them and made any corrections. Okay, we are going to start with looking at a plant cell. So please draw this. This is fundamental for a lot of what you learn in biology, is knowing the structure of a plant cell and an animal cell. Today we're talking about photosynthesis, so we are thinking about plant cells. Now, make sure you can label each part. So starting at the top left-hand corner, you've got the cell membrane. Look where the cell membrane is labelled. Your label has to be really careful if you're asked to do that in an exam. And the cell membrane controls the movement of substances in and out of the cell. Next one, we'll go anti-clockwise. So next one is the cytoplasm. Most chemical processes happen there. The next one is the nucleus, which contains genetic information. Now, all these parts of the cell on the left-hand side are found in plant and animal cells. So they are found in both. And that is a common question that comes up, asking you for a part of a cell that is found in both plant and animal. So make sure you know cell membrane, cytoplasm, and nucleus are all found in a plant cell, as well as animal cells. And then on the right hand side, we have got, starting with the chloroplast. So chloroplast contains a chemical called chlorophyll, which absorbs light for photosynthesis. There is also a vacuole, which contains cell sap, so that's labelled there. And then you've got the cell wall, so look where the label is. It's the thick part of the surrounding area of the plant cell, which strengthens the cell. Now, these three parts are examples of parts of the cell that are only found in plant cells, not animal cells. So please make sure you can name those as well. So please make sure you've drawn it and make sure you have labelled them. Okay, moving on. So you will have done this at Key Stage 3, but this also is a revision session, so you should be able to do this now. Please can you have a go at writing a word and symbol equation for photosynthesis? So I've put the words there for you. You need to put them in the right order. Use the pluses, use the arrow, see if you can work it out. And if you can you write a balanced symbol equation for this process as well? Now, there aren't that many equations to learn in biology, but photosynthesis is one of them. Another one is respiration, aerobic and anaerobic, and another one is fermentation. But there aren't that many, so please make sure you know them. You don't want to lose easy marks by not knowing these equations. Okay, so moving on, let's see how you've got on then. So this is the word equation here. Carbon dioxide plus water makes glucose and oxygen. Light and chlorophyll are really important. Light is needed for photosynthesis, but it isn't made of atoms, so it isn't part of the equation, but we can put it on the arrow there. And chlorophyll, as mentioned before, is the green pigment that makes chloroplast green, and that is what traps the light. So also really important. Now put on the last slide, you know, how do the plants get the carbon dioxide and the water? Well, the water is taken in by the roots, by osmosis. So that's another part of B1 that you need to know about. And carbon dioxide is taken in at the leaves through the stomata. And remember that the stomata are surrounded by guard cells and they can open and close. So they do open to let carbon dioxide in, but they can also close as well when they are dehydrated and they need to keep water. We will talk more about the products of glucose and oxygen, but obviously oxygen is released into the atmosphere and keeps us alive. And we will talk a little bit more in a couple of minutes about the few things that glucose is used for. Now, before we get on to the idea that this is an endothermic reaction, I just want to point out the symbol equation here at the bottom, particularly if you're doing the higher tier exam. Please make sure you know this equation. And as I will have said to you before, if you know the equation for photosynthesis, you know the equation for respiration, because you just swap the two products here to the other side for respiration and vice versa. Okay, now this is an endothermic reaction. So this is linked to chemistry, and an endothermic reaction is one that takes in energy. And if you think about it, it's taking in light energy from the sun for photosynthesis to take place. So it is an endothermic reaction. And opposite is respiration, which is an exothermic reaction. So a little bit of revision there on respiration as well. So if you remember that photosynthesis takes in energy to be able to do this process, so it's endothermic, and respiration gives out energy in the form of heat, therefore it's an exothermic reaction. Make sure you've written those down, make sure you learn them, please. Okay, moving on then. So I said we'd talk about what plants use glucose for. This is a very common question on exams, and one, if I'm honest, pupils can forget about. So please make sure you can describe ways by which plants use glucose. So plants photosynthesize so that they make glucose, so the glucose can be used for respiration. We respire as well. Plants respire. To get the glucose we need for respiration, we will eat food. Plants cannot eat food, so they have to make their own in photosynthesis. What they then do with the glucose from photosynthesis is that they use it for respiration. So, respiration is that chemical process by which glucose reacts with oxygen to release energy, water, and carbon dioxide. So please do not forget that plants respire too. Some of the glucose will be stored as starch, which is insoluble, and some will be stored as fats. And they can be used later if the plants need them, so that they will be there as a storage molecule, so they'll be there for energy. Now, others that are important for you to know is that glucose can be used to make new chemicals such as cellulose. So cellulose is a chemical that makes the cell walls, and it can also be used to produce amino acids to make proteins. Now, nitrates or minerals from the soil also help with that, but it's really key that the proteins are made, because that is what helps the plant to grow. Okay, so please remember, we've got respiration, we've got storage molecules, and then we've got the glucose being made to make other molecules such as cellulose and protein, which help the plants to grow. Okay, so jot those down, make sure you know examples and you can recall them for the exam. Okay, moving on. So photosynthesis and respiration, plants do both. Okay, I'm going to keep saying that because pupils often think of photosynthesis about plants and respiration about animals. Plants do both. So during the day, plants will photosynthesize because there is light available. Okay, and there will be some respiration going on. So during the day, photosynthesis will be taking place and some respiration. At night, photosynthesis will not be taking place. Okay, because there is no light, so just respiration will occur. Now the plant will have oxygen because it will have made it during photosynthesis, so that oxygen that it makes during photosynthesis can actually be used at night for respiration. So please remember that plants during the day photosynthesize and respire, and at night they will just respire because there is no sunlight. Okay, fantastic. Right. This is another required practical that you need to know for B1. So this is investigating the rate of photosynthesis using the equipment that is shown there. So this equipment here, we've got a lamp, we've got a thermometer, a beaker, that's a funnel upside down, a boiling tube, and some pondweed. So what you can do is you can measure either collect all the oxygen at the top there and work out the volume of oxygen given off per minute, or you can count the number of bubbles. Now in school, when I've done this in the past, we've counted the number of bubbles, but it's quite hard to do because they can be tiny or they can be clumped together. So it's often more accurate to actually work out the volume. So if you can see here at the top, the oxygen is being collected. The reason we use pondweed is because obviously plants give out oxygen, but pondweed grows in water, so you can physically see the bubbles. Whereas a plant that is just sat in a living room or sat in a classroom, you can't see the oxygen because it's just a gas going into a gas. Okay, so what we will do is that you would put a lamp, for example, 5 centimetres away. You'd let it settle for a few minutes, then you'd count the bubbles in a certain amount of time. Then you would move it further away and then further away and further away. Please remember to make this practical as reliable as possible. You do repeats. So remember, reliability equals repeats. So in an exam, if they ask you how to make a practical more reliable, you say that you would repeat each data point that you've taken or each practical that you have done. Okay, so you have to get that word repeat in there somewhere. And at each part, you would count how many bubbles, okay, and then you would see what happens as the lamp moves away. Now, I have put the method on the next slide, which is here, with some keywords missing out. I would suggest that you write this method out and make sure you know it. Often they put a 6-mark question on an exam and they ask you how you would test for the rate of photosynthesis, and you have to be able to describe exactly what is going on. So pause the screencast now, see if you can write this method and fill in the gaps. Okay, so number 1, set up the equipment as in the diagram. Number 2, leave for 5 minutes for the pondweed to acclimatise to the first light intensity. Number 3, count the number of oxygen bubbles given off in 1 minute. Number 4 is done for you. Number 5, move the lamp 20 centimetres further back. Number 6, leave for 5 minutes for the pondweed to acclimatise again. Number 7, count the number of oxygen bubbles given off in 1 minute. Get at least 3 readings at each distance so that an average can be taken. Number 8, repeat by moving the lamp away by 10 centimetre intervals until 100 centimetres is reached. Record all results in a results table. Okay, so please make sure you are referring back to the required practicals for B1 because there will be some questions on the exam papers. Okay, moving on. So that practical that we've just been talking about was about looking at light intensity and how light intensity has an impact on photosynthesis. Now, during that, everything else should have kept the same. So the temperature should have been kept the same. The amount of carbon dioxide should have kept the same. Otherwise, it is an unfair test. Okay, so please remember when you're doing these practicals that they might ask you about factors that have to be controlled. So when light is being investigated, the temperature and carbon dioxide need to be kept the same. The amount of pondweed used needs to be kept the same. The amount of water used needs to be kept the same. The same lamp should be used, etc., etc. Now we can also investigate other factors. So we can investigate the impact of carbon dioxide. So you can add different masses of sodium hydrogen carbonate powder and that releases CO2 in the atmosphere. Or you could investigate the impact of temperature. So you could place the apparatus in water baths at different temperatures. Now remember, if you're investigating temperature, everything else needs to be kept the same. So the light intensity, the carbon dioxide levels need to be kept the same. If you're investigating carbon dioxide, the light intensity and temperature need to be kept the same. So you only change one variable. Okay, excellent. Right, moving on. So the next part that we're going to talk about is only named on the higher tier. So if you know you're doing the foundation paper, you can stop watching now. Thank you for watching the screencast. We've covered the majority of photosynthesis within those sort of 15 minutes, so well done. If you are doing the higher paper, please watch on. Okay, you need to know what a limiting factor is. So when a process is affected by more than one factor, the one at the lowest level will limit the rate of the reaction. So what that means is photosynthesis is affected by temperature, light and carbon dioxide levels. If one of them is at a really low level or at a level that isn't efficient for photosynthesis, that will be its limiting factor. So for example, a plant is growing and it is a beautiful day. Okay, so there's absolutely loads of sunshine, and so there's lots of light on the plant. It's warm enough, so it's a nice warm day, but for whatever reason there isn't a lot of carbon dioxide in the atmosphere. Carbon dioxide would be the limiting factor. So it's the one that is at the lowest level, or the level that is too far away from what the plant needs. That is what we call a limiting factor. Okay, so the three limiting factors, as just mentioned, are light intensity, carbon dioxide concentration, and temperature. The first two graphs here that you may have to interpret in your exam, the first two graphs show light intensity against rate of photosynthesis and carbon dioxide against rate. Now, if we're describing them, remember what I always say, which is say what you see. So for this one, as the light intensity increases, the rate of photosynthesis increases, and then the rate plateaus. So even though you increase in the light intensity more and more and more, the rate of photosynthesis isn't getting any higher. And that is the same for carbon dioxide. Now, what is happening at this part of the graph and at this part of the graph is that here, light is the limiting factor. We definitely know light is the limiting factor, because if you increase the light, you increase the rate of photosynthesis. Now, what happens when the line plateaus is that either carbon dioxide or temperature becomes the limiting factor, because you're still increasing the light intensity, but nothing is happening. So at this part of the graph, light intensity is the limiting factor. At this part, it's either CO2 or temperature. Now, the same applies for this one. At this part of the graph, carbon dioxide is the limiting factor, but at this part, it is either light intensity or temperature. Okay, so it's the one that is causing the rate of photosynthesis not to increase anymore. Now, this graph is slightly different, and that is because we have to link it to enzymes. So after a certain temperature, enzymes will start to denature. Before a certain temperature, enzymes will be working really slowly. So this graph looks a little bit different, because this is to do with the enzymes and the fact that at low temperatures, enzymes work slow. At high temperatures, enzymes will be denatured, and at the correct temperature, they will be working as efficiently and effectively as possible. And remember, enzymes are needed for photosynthesis because enzymes help to speed up chemical reactions, and photosynthesis is a chemical reaction. Okay, so make sure you recognise those graphs, make sure you've drawn them and labelled them. Okay, moving on then. This is just a little summary. So interaction of limiting factors. When a process is affected by more than one factor, the one at the lowest level will limit the rate of the reaction. So what would happen if you increase the limiting factor, e.g. light available? So if light available was the limiting factor in the first place, and then you started increasing the amount of light, the rate will start to increase, and then, but at some point light will stop being the limiting factor, and now something else may become the limiting factor, such as carbon dioxide or temperature, which we will look at on the next graph. So have a look at this. We've got light intensity along the bottom, got rate of photosynthesis up the side. Have a go at answering these questions at the top. So pause the screencast, see if you can answer those 4 questions. When you've had a go, please unpause the screencast. Okay, so at A, what is the limiting factor? That is the light intensity. We can see that because if you increase the light intensity, the rate of photosynthesis goes up. What is the limiting factor at B? So at B, we've got loads of light, but we've got a cooler temperature. So that would be temperature. At C, lots of light again, a nice warm temperature. So the limiting factor is probably going to be carbon dioxide, but it still could be temperature. So it still could be a little bit cool. It might still need a bit of warmth. Okay, but it's definitely not light. And the last one, explain why at B, the light intensity no longer increases the rate of photosynthesis. And at B, it no longer increases the rate of photosynthesis because something else is limiting photosynthesis. Something else has become the limiting factor, and it's either temperature or carbon dioxide. Okay, right, remember this is on the higher tier, so it will be more likely to be on your paper, as it will be really testing whether you understand photosynthesis and limiting factors. Okay, moving on. This is something else as well that you will have to know for the higher paper. It's something called the inverse square law. So a little bit of maths within biology. So the rule is when you double the distance from the light source, the light intensity falls by a factor of 4. So we're talking now about those practicals. So this would probably be a question that would come up within a practical question, a required practical question in the exam. So this is the equation. If you're working out the relative light intensity, you do 1 divided by the distance from the light source squared. So you square this number first and then divide 1 by it. Now note here, this does not have units. You don't need to make any units up. It is just a comparison, and it is comparing the intensity of the light at a distance from the source when the source is placed at 1 meter. Okay, so it is just a comparison, so there is no need for any units. Okay, so here is an example. A group of students investigated the rate of photosynthesis of pondweed, placing the light source 0.5 meters from the plant. What was the relative light intensity at this position? So relative light intensity is 1 divided by distance from light squared. So here it is 0.5 squared. So if you square 0.5, you get 0.25. Divide that into 1, the answer is 4. And remember, this is a comparison, so there is no need for any units. Okay, moving on. Another one, this time the light is 20 centimetres away. Now, be careful, because this equation, the distance from the light needs to be in metres. So what I've done here is I've changed the centimetres into metres. So centimetres is 20, it is 0.2 metres. So what was the relative light intensity at this position? You need to do 1 divided by 0.2 squared. So you'll square that 0.2 first, divide it into 1, which will give you a relative light intensity of 25. Okay, so make sure you can use that equation, make sure you recognise where it is needed. But again, this is on the higher tier, so it would possibly be on the higher paper. Okay, right, so that is photosynthesis revision. We've covered the equations, we've covered the uses of glucose, we've also talked about photosynthesis versus respiration, limiting factors, and we've also talked about the inverse square law. So thank you for listening, thank you for watching. I hope it's been useful. Have a lovely day. Take care.